Hi friends, my name is 𝗵𝗎𝗻. I am studying second BIPC in TTWREIS Girls Forum. We are surrounded by beautiful nature. So, I would like to ask you one question about nature. Do you know the tallest and smallest plant in the world? Yes, the tallest plant is Eucalyptus globulus and the smallest plant is 𝗹𝗶𝗇. How they have grown? What made them to perform many functions to keep them alive? So, it all happens with the help of cell. The cell is the basic unit of life and building block of every living organism. You may understand that my topic is about cell cycle and the mitosis division. Let us discuss some of the subtopics. Introduction, plant cell, cell cycle, mitosis division, regulations of the cell cycle. If you see the history of the cell evolution, it was first discovered by Robert Hooke in 1665. The process of cell division under microscope was discovered by a German botanist Hugo von Mohl in 1835 and cell cycle was discovered by Walter Fleming in 1882. Now we are going to discuss about types of cell. All organisms, even largest, start their life from a single cell. But you may be wonder how a single cell can form a complex body structure. How a single cell would form you? How a single cell we cannot see with our naked eye can form a complex plant body. So, it is due to the presence of some specialized characters to cell are growth and reproduction. Cell reproduce by dividing into two daughter cells and the progeny is genetically identical to the parental cell and number of chromosomes are maintained. Based on the cell, there are two types of organisms, unicellular organisms and multicellular organisms. Uni means single, cellular means cell. The organisms which are made up of single cell are known as unicellular organism. The best example is Chlamydomonas. The next one is multicellular organisms. Multi means many, cellular means cell. The organisms which are made up of more than single cell are known as multicellular organisms. The best example is tree and grass. Now we are going to discuss a few points about the plant cell. Plant body is made up of millions of cells. Every living organism is made up of cell. It has a protective layer called cell membrane. But especially for plant cell, there is an extra protective layer called as cell wall, and the cell wall is made up of cellulose, pectin, lignin, which is absent in animal cell. In animal cell, we observe centrioles which help in cell division and are absent in plant cells. How many times a cell will divide? For example, we take in plants meristematic tissues which divide continuously throughout their cell life cycle. But there are certain cells which form and undergo differentiation, means they become matured to perform specific job. The cyclic changes that takes place in a cell to perform divide, this is called as cell cycle. Cell cycle refers to the series of events like DNA replication, division of cytoplasm. Finally, it divides into two daughter cells. Now, phases of cell cycle. This process is more straightforward in prokaryotic organisms than eukaryotic organisms. Pro means false, karyon means nucleus, false nucleus is present in prokaryotic organisms. Means nucleus is present but it is not enveloped by the nuclear membrane. The best example is bacteria. The next one is eukaryotic organisms. Eu means true, karyon means nucleus. True nucleus is present in eukaryotic organisms, means nucleus is enveloped by the nuclear membrane. The best example is plant cell and animal cell. When we talk about the cell cycle, there is a preparation phase and active division phase. The cyclic changes takes place inside the cell in a regular manner. The best example is yeast, which can complete its cell cycle within 90 minutes. It is divided into two subphases. They are interphase and mitosis phase. Now we are going to discuss about interphase. Interphase is also known as non-dividing phase. It is a type of resting stage in which the cell is present in resting time. So scientists thought that interphase is a resting stage. So any activity does not take place during the interphase. But later they found that interphase is a preparation phase in which preparation takes place for the next stage. It takes longest part in the cell cycle. Its duration is about 23 hours. It is divided into three subphases: gap 1 phase, synthesis phase, gap 2 phase. Now we are going to discuss every phase one by one. The first one is gap 1 phase. Gap 1 phase is the end of the previous M phase until the beginning of DNA synthesis. During this phase, some biosynthetic activities takes place for the next stage. After gap 1 phase, there is synthesis phase. In synthesis phase, DNA replication takes place. You got the point that gap 1 is preparing some materials required for the DNA replication. But you may be confused why the materials are required in synthesis phase, but why they are prepared in gap 1 phase. For example, we take human cell. It wants to divide into two daughter cell. It has 46 chromosomes. It should give complete information to two daughter cell. But the complete information is present on 46 chromosomes. How it can give? For example, there is a page of information. It should give to two daughter cell. Without tearing the paper, the information should give in. By tearing the paper, it can give, but it is not a complete information. For this, copy is required, can be done in the next phase is the synthesis phase by DNA replication. The biosynthetic activities like protein synthesis, RNA synthesis, adenosine triphosphate, in short ATP synthesis takes place. When we can observe a cell at the beginning of the gap 1 phase, it will be in small size, but at the end of the gap 1 phase, it will increases its size. Now the next phase is the synthesis phase. Synthesis phase begins when DNA synthesis and all of the chromosomes have been replicated. So replication means a cell divides into two daughter cells must contain the same genetic information. DNA replication is a semi-conservative process. Each strand in DNA double helix acts as a template for the synthesis of the new strand is the complementary strand. Each formed DNA double helix consists one old strand, one new strand. If it is an animal cell, centrioles also divide, but it is not applicable for plant cell because centrioles are absent in the plant cell. Histone protein synthesis can be takes place in lower eukaryotes 50%, in gap 1 phase 50% in synthesis phase, but in higher eukaryotes 100% synthesis can be takes place in synthesis phase. So we can consider that histone protein synthesis can be takes place during the synthesis phase. Now the last subphase is the gap 2 phase. Gap 2 phase is similar to the gap 1 phase. So after gap 1 phase there is synthesis phase. In synthesis phase DNA replication can be takes place. So gap 1 is preparing materials required for the DNA replication. Like after gap 2 phase there is mitosis division, in which division can be takes place. So gap 2 is preparing for the cell division. When a cell have to divide, nucleus have to divide, new organelles should formed, chromosomes get separated. They are separated with the help of spindle fibers, and spindle fibers are formed by protein tubulin. So you got the point that protein synthesis can be takes place. To move a chromosome from equatorial plate to pole require 30 ATP for a single chromosome. So ATP synthesis can be takes place. New organelles should formed. So plasma membrane proteins also required. During gap 2 phase, protein synthesis, RNA synthesis, ATP synthesis going to increases. Now the cell is ready to divide is the mitosis division. Now we are going to discuss about the mitosis division. Mitos means thread, osis means state. Active spindle fiber formation can be takes place. This process explained by Strasburger in plants. The term mitosis coined by Fleming. It takes place in somatic cell. If it is an animal cell, it takes place in diploid somatic cell. In plant cell, it takes place in haploid and diploid cell. A haploid plant body produces gametes by mitosis, and the plant body is gametophytic. Zygote also divide by mitosis and produce diploid sporophytic plant body. During mitosis division, the number of chromosomes in parental cell is equal to the daughter cell. So it is also known as equational division. It takes shortest part in the cell cycle. Its duration is about 1 hour. It is divided into 2 substages: karyokinesis, cytokinesis. Karyon means nucleus, kinesis means division. Nucleus division in mitosis can be takes place in 4 subphases. They are prophase, metaphase, anaphase, telophase. Now we are going to discuss every phase one by one. So prophase follows S and G2 phases of the interphase. During S and G2 phases, new DNA molecules are formed, but they are not distinct, they are intertwined. During this phase, initiation of condensation of chromatin materials, and the chromatin materials entangled during the process of chromosomal condensation. Nuclear membrane, endoplasmic reticulum, Golgi complex membranes dissociate, and centrioles moves to the opposite poles, and chromatin material condenses. So from centrioles some fibers arises. Some fibers are long and some fibers are short. The long fibers move from one pole to the another pole and help in the attachment of the chromosome are known as spindle fibers. And the small fibers are called asters. There are two asters in a cell known as Mitosis Division, in which asters are absent in the plant cell because centrioles are absent in the plant cell. It is known as Mitosis Division. Now, the next phase is the metaphase. During this phase, complete disintegration of nuclear envelope occurs, and also we clearly observe the chromosomes under the microscope. So, this phase is favorable to study the morphology of the chromosome. The metaphase chromosomes with the two chromatids held at a point is known as centromere. At centromere, some disc-shaped structure is present, is called as kinetochore, and the kinetochore help in the attachment of the spindle fiber. You can observe two types of spindle fibers here. Here, some spindle fibers help in the attachment of the chromosome, but some spindle fibers not help in the attachment of chromosome. Which help in the attachment of the chromosome are known as chromosomal spindle fibers, and there is a discontinuity due to the attachment of the chromosome, so it is also known as discontinuous fibers. Which not help in the attachment of chromosome are known as interpolar fibers because they move from one pole to the another pole. There is no any disturbance due to attachment of the chromosome. So, it is also known as continuous fiber. Here you can observe one of the chromatid of the chromosome is connected to the kinetochore or spindle fiber in one pole, and another chromatid of the chromosome is connected to the spindle fiber of opposite pole. This makes the chromosomes to present in a equal plane is known as equatorial plate or metaphase plate. They get splitted in the next phase is the anaphase. So, during this phase, we discussed above. During metaphase, the chromosomes present in equal plane is a metaphase plate. They get splitted in this phase. The chromatids moves to the opposite poles and they becomes the chromosomes of future daughter nuclei. Between the splitted centromere, there is an additional fiber. It is known as interzonal fiber. For the movement of chromosome from equatorial plate to pole, some forces are responsible, like contraction of spindle fiber, stretching of interzonal fiber. We discussed about that. To move chromosome from equatorial plate to pole requires 30 adenosine triphosphate. So, you can consider that ATP is also participating for the movement of chromosome from equatorial plate to the pole. Now, the final stage of the karyokinesis is the telophase, which is completely reversed to the prophase. So, during prophase, nuclear membrane, Golgi complex, endoplasmic reticulum membranes dissociate, centrioles moves to the opposite poles, and chromatin material condenses, which is completely reversed in the telophase. In the telophase, chromatin materials decondenses, loses their individuality and form masses at the either poles. Nuclear membrane, endoplasmic reticulum, Golgi complex membranes reappears. Finally, the nucleus is divided into two nuclei. In the cell is the nucleus. So, we observe that the karyokinesis is divided in four subphases. They are prophase, metaphase, anaphase and telophase. So during prophase, the chromosomes are condensed and they get present in equal plane in the metaphase. They get splitted in the anaphase, and finally the nucleus is divided by telophase. Now the nucleus division is completed. Now the cytoplasm division is the cytokinesis. Cytokinesis is different in plants and animals. In animals, it takes place on the plasma membrane, depends and divides into two daughter cells. The cell is divided into two daughter cells in the animal, but in plants it is different because due to presence of hard cell wall, it takes place at the center of the cell by forming a cell plate. It has the deposition of calcium and magnesium, so it behaves as a middle lamella. It moves to the opposite poles. Finally, plant cell is also divided into two daughter cells. Now, the cell is divided into two daughter cells. Again, the daughter cell will again enter into the interphase and continue same process, and it also divided into two daughter cells. So it is a complete cycle in our life and every living organism. In some organisms, cytokinesis does not follow the karyokinesis, results in multinucleate condition, leads to the formation of syncytium. The best example is liquid endosperm of coconut. So of all this we can observe the best example is onion root tip division. For this we want to take an onion and kept in a test tube containing water for 2 to 3 days, and then some roots will arises. From the down part of the onion. So they are onion roots. We want to take tip of the root because division of multiplication is very fast at the tip of the root. We want to take this tip of the part and observe under the microscope. We clearly observe them all prophase, metaphase, anaphase, telophase in karyokinesis and cytokinesis, the cell division completely by this process. Now the division is completed. We are going to discuss that. Okay, the two daughter cells are formed, but the two daughter cells differentiated to perform specific jobs. They again enter into the cell cycle in the interphase to regain their size. Then they enter into a stage known as G0 phase. In this phase, cell perform all activities except the cell division. So it is also called as inactive for division or quiescent phase. It can be explained by 2 to 3 ways, like the cell enters into the cell cycle, regains its size and stay for some time and move out of the cell cycle. This process is known as differentiation. The cell will not enter again into the cell cycle. For exception, the parenchymatous cells, which help the formation of the secondary growth in the dichostem, they again enter into the cell cycle, become meristematic and helps the formation of the cambium. So the again entering into the cell cycle, this process is known as de-differentiation. If the cell is completed all the activities, again enter into the cell cycle, out of the cell cycle, this process is known as redifferentiation. Finally, regulations of the cell cycle. When we are learning a topic, for example, cell cycle is a very complicated process. So there are some checkpoints to check whether all activities are taking place in a proper manner or not. When we are discussing a topic, it should be useful to us or environment. For example, we are traveling in the road. Suddenly accident will occur, our body is ruptured. But after a few days, we are recovered. By which division? It is mitosis division. It helps in repair, regeneration, growth of the body and healing the injury. Finally, I conclude that cell division is a very important process in living organism to continue their life cycle. The mitotic division, the number of chromosomes not increases and decreases, remains constant. Cell division is very fast at the tip of the root and the shoot, and the mitotic division in the meristematic cells of the plants help in the continuous growth of the plant. So, I am thankful to my guide teacher 𝗧 Rajita madam and my principal madam 𝗧 madam and to all TSAT programmers, and a special thanks to Secretary sir and society. Thank you to one and all.